Our next speaker is Yang Den from UC Berkeley, and she'll be talking about neural circuits for sleep-wake control. Thank you.
So mammals and birds have three distinct brain states: wakefulness, non-REM sleep, and REM sleep. During wakefulness, the EEG is what they call desynchronized. You see this high-frequency, small amplitude activity, and the EMG shows high level of muscle tone. And this is a state when we have vivid sensation of the external world. During non-REM sleep, this is also called slow wave sleep because the EEG is dominated by this large slow wave activity. The EMG shows reduced muscle tone, and we have either dull or absence of sensation. And during REM sleep, so this was only discovered about 60 years ago in the 1950s. It's also called paradoxical sleep. It's paradoxical in the sense that the EEG is very similar to wakefulness, but the EMG shows a complete lack of muscle tone. And this is a state when we have vivid dreams, and the idea is that maybe this so-called REM atonia can prevent us from acting out our dreams. So our interest is to figure out what neural mechanisms control sleep and wakefulness. Now, over the last century or so, the work from many labs has identified many brain areas that are involved in sleep-wake control. These include the hypothalamus, brainstem, basal forebrain. This is from a review by Saper at Harvard. And you can see that it includes many of the very well-known neuromodulatory circuits, the monoaminergic neurons, cholinergic neurons, peptides, and so forth. But in each of these areas, there are also many glutamatergic and GABAergic cells, just like in the rest part of the brain. But the circuits are not very well understood. So what we're hoping to do is to try to understand the circuit. About 4 years ago, we started working on the basal forebrain. Actually, Mike Gourd was the first one in my lab looking at basal forebrain modulation of visual processing. But then that sort of led us to this whole sleep-wake regulation. And then about 2 years ago, we started working on the brainstem. And just earlier this year, we started working on the hypothalamus. But today, I'm going to focus on the basal forebrain and brainstem. So what do we know about the basal forebrain? We know that lesions in this area, in some cases, cause insomnia, but in other cases, increase the slow EEG activity. So as you remember, this is sort of the sleepy state, right? So that suggests that this area is important for both wakefulness and sleep. In terms of cell types, there are three major cell types: cholinergic neurons, about 10 to 20%, glutamatergic neurons, 20%, GABAergic neurons, by far the majority. So the questions that we have been focusing on are two major questions. One is the functional property of each cell type. And for that, we'd like to know how does the activity of each cell type affect brain state, and also what are the firing patterns of these cells in different brain states. And we also like to know the synaptic interactions between cell types. So we started out with the cholinergic neurons, partly because we already know about these cells quite a bit, and also Mike has looked at these cells, how they affect visual processing. So we either use the mouse generated by Guoping Feng's lab here, or the Chat-Cree mouse, but injected with the virus expressing channelodopsin in a Cree-dependent manner. And so you can see that in this case, YP channelodopsin is expressed specifically in the cholinergic cells. And if we patch onto one of these cells in vitro, you can see that each blue light pulse, this blue dot here, can evoke a spiking reliably. And then to look at how it affects brain state, we implanted optic fiber into the basal forebrain and also recording EEG and EMG so that we can determine the brain state. And here are three example trials. So the laser was applied for 10 seconds during this blue shading period. And you can see that in each trial, during laser stimulation, the EEG is desynchronized, and then the EMG shows an increase in the activity. So that suggests that the stimulation was enhancing wakefulness. So this is just summarizing the result. So laser stimulation decreased non-REM sleep, increased wakefulness, and didn't change REM sleep that much. And this is the time course of the effect. So in these experiments, we stimulated for 60 seconds, but you can see that basically within the first couple seconds of stimulation, there's a very obvious change in the probability of the different brain states. So the activity of these cholinergic neurons can affect brain state. But of course, the other question is, what are the natural firing patterns of these cells? Because normally, I think one of the speakers made a point this morning that normally you don't have the blue laser in the brain to activate these cells. So we needed to know that these cells were naturally active at the right time. And now, like I said, there are 3 types of neurons in the basal forebrain. The cholinergic neurons are really the minority. So to identify these cells, we use optoid recording. So this is an optoid we made in the lab, slightly modified from the design, published by Carl Dijsselra's lab. There's an optic fiber surrounded by several bundles of either stereotrodes or tetrodes, depending on the brain area. And this is just to show that we implant the optoid into the basal forebrain. And then once we implanted the optoid, we looked for neurons that are reliably driven by the laser pulses. So here, each blue dot is a 5 millisecond laser. And you can see that here's a unit that's reliably driven by it. And this shows multiple trials of the laser pulse train, PSTH. And this shows the distribution of the spike latency. So for cholinergic neurons, the latency is about 6 milliseconds. But for many other cell types, the latency is a lot shorter, 2 or 3 milliseconds. So when we see a unit like this, we're fairly confident that this is a neuron that expresses channelrhodopsin, and therefore, in this particular mouse, it should be a cholinergic cell. And this shows the firing rate of this cell recorded over about 2 hours, I think. This upper trace is the EEG power ratio between theta and delta. So theta is about 8 hertz, delta is 1 to 4 hertz. So this ratio is high whenever the animal is either awake or in REM sleep. The second one is EMG power, and it's only high when the animal is awake. And the third one is the firing rate of that particular cell. So you can see that this neuron was firing at high rate during both wakefulness, this red region, and also REM sleep, this blue region. But it's essentially shut down during non-REM sleep. And so to quantify the firing rates of these neurons, we plotted each cell, each circle here, in this 2-dimensional plot. So the horizontal axis is the relative firing rates between the wake and non-REM sleep states. So it's the difference divided by the sum. And the vertical is REM versus non-REM. So you can see that all 12 identified cholinergic cells fall in this upper right quadrant, indicating that they're more active during both wake and REM sleep. So this gray shading region indicates the firing rate modulation being less than 2-fold. So if it's within this region, we consider it weakly modulated. It's somewhat of an arbitrary border. But you can see that all the 12 cholinergic neurons are very strongly modulated because they're all outside of this gray region. So now I have to say that way before we did this experiment, this was already known. So this is a paper published by Barbara Jones' lab. And you can see that these neurons were active both during wake and PS, which is just another name for REM sleep, but they're shut down during slow wave sleep. Now, in this experiment, what they did was to make juxtacellular recording and labeling in head-fixed rats. So they have to train each rat for 10 days, 2 weeks, so that they can sleep when they're head-fixed, because this is really only practical in a head-fixed animal. And then after that, they sacrificed the animal and made immunohistochemical identification of the cholinergic cells. So the yield of this experiment is incredibly low. So in this particular paper, they only had 5 identified neurons. So basically what we have done is to use this sort of more modern technique enabled by optogenetics to reproduce their finding, but with a higher throughput. So once we validated our technique, we went on to look at the other cell types. The next one is the glutamatergic cells. And so we use the same trick, looking for the driven units in the VGLUT2 Cree mice. And then here's an example unit, and you can see that this neuron is also more active during wake and REM compared to non-REM. And this is a summary of 31 glutamatergic cells. And you can see that most of them are in the upper right quadrant. And then there's some of them that are in this gray region, meaning that they're weakly modulated. And so what about optogenetic activation of these glutamatergic cells? So I kind of wanted to show the video of this because it's quite dramatic. So the upper trace is the EMG power, and the second one is the power spectrum of the EEG. So when they're asleep, you see this broadband low frequency activity. This is the brain state classified based on these recordings, and the laser is indicated here and also by the blue dot here. And you can see that the animal was in non-REM sleep, but the moment we turn on the laser, he started running around, very excited. And then even after we turn off the laser, he's still pretty active for a while. And then here's just another trial. So again, he was in non-REM sleep because of the broadband low-frequency activity. And then the laser is turned on, and he started running. In fact, it looks to us that he's pretty agitated, not just waking up, but very active. So this is a summary of the result. So again, we saw decrease in non-REM, increase awake, not much change in REM. And this is the time course. And so if you compare to the cholinergic neuron activation, this one wakes the animal up, is approaching 100% efficiency. And also the effect seems to be much longer lasting compared to cholinergic activation. So I just want to make the point here that in the basal forebrain, the cholinergic neurons are much more famous compared to the lowly glutamatergic cells. But in fact, the behavior modulation is stronger for the glutamatergic cells. So and then we moved on to the GABAergic cells. And previous literature already suggests that the GABAergic neurons are diverse. So we went for the different subtypes of GABAergic cells, like Andrews was talking about this morning. We also looked at the parvalbumin positive GABAergic cells. And so here's a summary of their firing pattern. Many of them in the upper right quadrant. Some of them are in the upper left. So these are the cells that are REM active but not wake active. But many of them are weakly modulated. And compared to the cholinergic and glutamatergic cells, these neurons also fire at much higher rates across all the brain states. So that's why they're the fast spiking neurons, as in the hippocampus and the cortex. And then if we activate the PV neurons, we again saw decrease in non-REM, increase awake, and no change in REM. And then we went on to the somatostatin positive GABAergic cells. And so for these, if we activate them, we actually saw the opposite effect, which is an increase of non-REM, decrease awake, and no change in REM. And then when we recorded the activity from these neurons, some of them are actually sleep active. So here is a pair of neurons that were simultaneously recorded. Both of them are somatostatin positive. And you can see that both of them actually fire more during non-REM sleep, slow wave sleep, compared to wakefulness. And one of them fired even more during REM state. So this is a summary of these two neurons across the three brain states. So at least we found some sleep active neurons in the somatostatin population. And this is a summary of all the somatostatin neurons. So now I showed some sleep active neurons, which are here, but there are also others that are just similar to the other cell types that we have looked at. So they're wake active. And you also see some cells in the second and the fourth quadrant as well. So these neurons are very diverse. And this is just a comparison. So I'm putting all the 4 cell types that we have looked at up together. So this is by far the most diverse. But the important thing here is that all of these cell types, the somatostatin neurons, are the only ones where we found strongly sleep active neurons. And the optogenetic effect is also sleep promoting. So that was the first question. And just a quick summary, we found that cholinergic, glutamatergic, and PV GABAergic neurons, they're all wake and REM active. And if we activate them, we promote wakefulness. But the somatostatin neurons, some of them are sleep active. And if we activate them as a population, it increases non-REM sleep. And the next question is the synaptic interaction between cell types. So first, we looked at the local connections. And to do this, we went to brain slices. So here's an example where we use a double transgenic mouse with channelrhodopsin expressed in the cholinergic cells and TD tomato in the PV positive cells. And so if we patch onto the green or the red cell, we can see that the cholinergic cells are kind of regular spiking, and the PV cells are fast spiking. And then if we flash on the blue light, actually this is the wrong color, but it's a blue laser pulse, we can evoke spiking in the cholinergic cell, but then the synaptic response in the PV cell. So this is this EPSP was unaffected by CNQX, so meaning that this is not mediated by glutamate, but it's completely wiped out by the two drugs that together completely block the nicotinic ACH receptors. So based on that, we concluded that the cholinergic cells excite the PV cells primarily through the nicotinic ACH receptors. And in fact, people have published papers to show that if you really activate these cells with a prolonged activation, you might see some muscarinic components as well. But with a single pulse, it seems to be mostly nicotinic. And this is a busy slide. I don't mean for you to go through all of them, but just to show that we have looked at all the pairwise interactions that we have the mice for. So the top is the presynaptic neurons, so we have each of the 4 cell types, and the bottom is the postsynaptic cell type. So we have a couple of them that are missing just because we didn't have the right mice for those experiments. But so based on this kind of data, here's a summary of the local circuit across the 4 cell types. Now, these 3 light circles are the wake-promoting neurons, and then this one is the sleep-promoting neuron. And so red is excitatory and blue is inhibitory. So it seems like there are 2 things that sort of jumped out at us. One of them is that the wake-promoting neurons seem to be organized hierarchically in the excitation chain, in the sense that the glutamatergic neurons excite the cholinergic and also the PV cells. Cholinergic cells also excite the PV cells. But the feedback is either absent, so PV cells seem to be avoiding the cholinergic cells. But the cholinergic cells provide a tiny bit of excitation to the glutamatergic cells, but it's primarily inhibition. This inhibition is just way, much stronger than excitation. So based on this, we think that the behavioral effect that we saw also makes sense because, like I mentioned, if we activate the glutamatergic cells, we saw this very dramatic wake-promoting effect. And we think that's because when we activated these cells, this also recruits the cholinergic and the PV cells. So both of these would separately promote wakefulness. But if we activate the PV cells, which we only saw a weak wake-promoting effect, and that's because these are the only cells that are activated, because these guys don't really recruit the other cell types. The other thing is that the sleep-promoting neurons, somatostatin neurons, they actually provide inhibition to all 3 types of wake-promoting neurons. And this is different from the PV cells because, like I mentioned, PV cells actually don't really innervate the cholinergic cells. They innervate the glutamatergic cells just a tiny bit, extremely weakly. But the somatostatin neurons provide very strong inhibition. And we think that this broad inhibition might be important for the sleep-promoting effect. And so of course, in addition to the local connections, the long-range connections are also important. And it turns out in the basal forebrain, all the 4 cell types that we looked at, they're not local. They all project out to other brain areas, and they also receive inputs from other brain areas. And so here I'm just going to quickly show you that we mapped these long-range connections. So in this case, we just injected the fluorescent tracer in each of the cell types, and then just to see where their axons go. And so you can see that the cholinergic cells are very special because they project big time to the cortex. But the other 3 cell types, they project to the hypothalamus, thalamus, and so forth, and maybe a little bit of the brainstem, but they really don't go to the cortex that much. And this is just the input. So for this, we use the rabies virus that was mentioned this morning, and Li Chun's lab is the one that helped us to get this to work. So we targeted each of the postsynaptic cell types, and then each red dot here represents the rabies virus labeled presynaptic cell. And again, you can see there's this very broad distribution of the presynaptic structures that feed to the basal forebrain. And I think that there was a discussion this morning that using rabies, we initially hoped to fish out a few sort of interesting areas, but we just ended up with so many, so it's very hard to figure out where to target next. Okay, so that's the first part of my talk, and now I'm going to tell you another story. Hopefully I can go through quickly. What controls REM sleep? Now in the first part, when we went through the different cell types in the basal forebrain, we see wakefulness or non-REM sleep, but nothing changes REM sleep. So what's going on there? Now it turns out that people have tried to identify the areas important for REM sleep by doing transaction studies. So these were pioneered by Juve in the early 1960s and later done by many others. And so this is a review by Jerry Siegel. summarizing the main results. Basically, if you make a cut at A or B, then you see normal REM atonia, this sort of muscle paralysis from time to time. But in the forebrain region, more rostral to this cut, there's non-REM and possibly wake, but there's no sign of REM sleep. But if you make a cut at C, then you see REM in the forebrain, but there's no spinally mediated atonia. So based on these experiments, people think that the region between A, B, and C contains the key circuits for REM sleep. And people have also done some cutting at D or E to show that the communications between the pons and the medulla might be important for REM generation. So the question is, what neurons are important for REM? Because in this region, there are neurons mediating many other functions. So that's why we moved to this part of the brain. So to identify potential neurons that are important for REM sleep, the standard paradigm has been doing CFOS staining. So this is a study done by the Lupi group in Lyon. So what they did was to first do a REM sleep deprivation. Anytime the animal goes into REM sleep, you wake them up. You do this for a few hours, and then you leave the animal alone. So that causes a REM sleep rebound. And then after about 2 hours of the rebound, you sacrifice the animal and you stain for the immediate early gene, CFOS. So here is a coronal section at this level. And now if you zoom in to this sort of ventral medulla region, you see the picture here. Basically, they found a large number of GABAergic neurons in the ventral medulla that became CFOS positive after REM sleep rebound. So that suggests that these neurons are activated during REM sleep, and so perhaps they're important. So the questions that we asked were, one, does the activity of these neurons cause REM sleep? Because the CFOS experiments suggest correlation, but it's not causality. And secondly, actually, we don't really know if all of these neurons are really specifically activated during REM. And that's because after REM deprivation, right, the animal spends about 40% of the time in REM sleep as compared to the normal 10% of the time. So it's a huge increase, fourfold increase, but it's not 100%, right? So there's 60% of the time the animal is in who knows what brain state. So the CFOS positive neurons are not necessarily all REM active. So we like to record from these cells. And then, of course, if we want to study the circuit, we like to know these cells' downstream targets and also their inputs. So to look at the causal relationship, we express channelrhodopsin in these cells using the GAT2 Cree mice. So you can see the expression in this region. And we implanted an optic fiber to activate these cells. And this is a movie showing the effect of activation. It's speeded up 10 times. So this is EMG power. This is the EEG spectrogram. And then so the animal was awake even before we turned on the laser. And actually, laser didn't seem to be changing things too much. So he's still awake, and then the EEG is still desynchronized. But in the second trial, he was actually in non-REM sleep. And you can see that the EMG is low, and then the EEG is dominated by this broadband low-frequency activity. And then the laser is going to come on, and you see a very immediate change of the EEG power spectrum. So now it's desynchronized, and you see this very clear theta band. So the theta band is very characteristic of REM sleep. But the EMG is still low, right? So that's how we know that he's in REM sleep rather than awake. And so there's a third trial, but I'm going to skip it because I'm running out of time. So this is a summary of that experiment over a longer period of time. And so the laser was applied for 2 minutes in each trial, and it was applied once every 15 to 25 minutes. So these blue shading, the laser period. And you can see that, so EEG power spectrum, EMG, and this is the color-coded brain state. So this light blue indicates the REM period, and you can see that 3 out of the 4 trials we turned on the laser, there was a REM period. But one of the trials, when the laser fell on the awake state, actually nothing happened. So this is a summary of all the trials that we got out of 6 mice, aligned by the time of laser stimulation. And you can see that during this 2-minute laser stimulation, this bar here, there's a lot of blue, right? So that suggests that there's increase in REM. This is the probability of each of the 3 brain states. And you can see that as soon as we turn on the laser, the non-REM sleep, there's a very dramatic decrease. And there's a complementary increase of REM sleep. Now, you might also notice that there's sort of a gradual increase of wakefulness, this purple line. Now, we think that this is actually a secondary effect of increasing REM, because in rodents, following REM, they usually wake up. They don't go back to non-REM sleep, and I'll come back to that point. So these complementary changes between the non-REM and REM suggest that maybe we're just really converting non-REM into REM. And so if we look at all the trials where the laser fell on non-REM state, and that's why the non-REM probability is 1 right before 0, then you see this even more dramatic increase of REM. It's 80 to 90% of the trials. But if you look at the trials where the laser fell on the awake state, then there's not much change. So that was the probability of each brain state. And now we also looked at the transition probability between each pair of brain states. So here is the transition from non-REM to REM. And so each bar is the transition probability within a 20-second period. So you can see that before laser, the transition probability is very low. But during the laser period, there's a dramatic increase of this probability. Now, if we look at the transition from REM to wake or non-REM to wake, so these are the only ways that we can get wakefulness. We can trigger wakefulness, right? You can see that this transition probability is actually reduced during laser, consistent with the idea that the laser is trying to promote REM. And the probability from non-REM to wake, actually there was no significant difference. Now you see these sort of tall bars near the end of the trial. So these have extremely large error bars, and that's because at this point, most of the non-REM have converted into REM. So we don't have many trials where the animal was still in non-REM here. But anyway, so this is not sufficient to give rise to the increase in wakefulness. And that's why I said the wakefulness was the secondary effect. The final thing I wanted to point out is that from wake to REM, this normally does not happen. It doesn't happen in normal animals. It only happens a lot in narcoleptic animals. And so you can see that even though the laser was really promoting this normal transition, but it didn't promote this narcoleptic transition. Okay, so these are the controls. And so if we only express YIP without channelrhodopsin in these GABAergic cells, laser didn't do anything. And if we express channelrhodopsin in the glutamatergic cells and do the same laser stimulation, we saw a very dramatic increase of wakefulness, right? In fact, 10 seconds of laser induced wakefulness up to 90%. So it seems like the REM promoting effect was specific for the GABAergic neurons and not for the glutamatergic cells. So that was for induction of REM. And we also looked at the maintenance of REM sleep. And for that, we applied a closed-loop stimulation protocol. So we monitored the brain state using EEG and EMG and made an online determination. And we only turn on the laser after the animal spontaneously enters into REM. So we skip this induction part and only try to maintain the REM sleep. And then the laser stayed on for as long as the REM episode lasted. And so here is an example experiment, and you can see that, so we actually apply laser stimulation in 50% of the REM episodes randomly. So you can see that there are some trials, REM, we didn't turn on the laser, but in other trials we did. And in the trials we turn on the laser, the REM duration seems to be longer. And this is a summary of the results. So each pair of dots is the data from one mouse. So there's 6 mice here. And you can see that basically laser doubled the duration of REM for the CHR2 mice, but not for the YIP control. And if we use arch or halorhodopsin to try to shut down these cells, we actually saw a shortening of REM, but that's not seen in the YIP. So it's green here because we use a green laser for this experiment. Okay, so here's a summary. So activating these cells can induce non-REM to REM transition. In our data, it's actually happening in 94% of the cases. And it also doubled the duration. And next, we asked whether these neurons are specifically active during REM. And for that, we use the same trick, optical recording. And so here is an example cell. So this is the firing rate of the cell. And you can just focus on the color-coded brain state. So whenever the animal enters into REM, you can see that this neuron always fired at high rate. But during non-REM sleep, the firing rate is pretty consistently low. But during wakefulness, this purple, the firing rate was actually variable. And in fact, we took a closer look at the data, and we noticed that these neurons fire during wakefulness, mostly when they're grooming or eating, sort of relaxed, enjoying themselves kind of mode, but not when they're running. So this is the mean firing rates of this cell over the 3 brain states. Is most active during REM. And this is a summary of 20 identified GABAergic cells. And this is the same data, except that we normalize the firing rate of each cell by its maximum. So black is the maximum firing rate. And you can see that all 20 units had the maximum firing rate during REM. And so this is the mean firing rate during the transition. So during the non-REM to REM transition, you see that the firing rates of these cells actually started going up about 20 seconds before the actual transition. And then the firing rate stayed high during REM, and then it dropped abruptly right at the end of REM. So we think that this kind of firing pattern allows these cells to be both responsible for the induction and maintenance of REM sleep. Okay, so these cells are most active during REM. And so what about their downstream targets? So we know that these cells project both rostrally to the pons and caudally to the spinal cord. In this experiment, we injected 2 tracers, retro-beads, green, and fluorogold red, into these 2 locations. And when we looked at this ventral medulla region, we see both the red and the green labeled cells, but there are very, very few cells that show the double labeling. It's basically about 1% of the cells. So we think that these 2 projection pathways came from largely separate cells. And then we took a closer look at this projection to the pons, because as I mentioned, based on early lesion studies, the pons is very important for REM sleep. And so you can see these green fibers kind of go into the pons. In this case, red is cholinergic staining, and in this case, red is labeling for the locus coeruleus. So basically, these fibers kind of project to all of these areas. Now, in this experiment, what we did was to express tyrosine in these cells, but then we activated their axon fibers in the pons. And you can see that for the induction experiment, we also saw a drop of non-REM and increase of REM, basically very similar to the effect of cell body stimulation. And for the closed loop experiment, we again saw this doubling of REM sleep duration. So we think that based on these experiments, we concluded that the activation of the neurons projecting to the pons is sufficient for REM induction. Now, we're not saying it's only the fibers we're activating with laser, because there could be antidromic propagation and collaterals. But since these neurons are separate from the neurons projecting to the spinal cord, we think that we're not activating the cells that project to the spinal cord. And then finally, the question about inputs. So I'm not going to show you any data because this is very much ongoing work, but it's very important to figure out who provides the input to these cells. And then so summary of this part of the talk, we show that activating these ventral medulla GABAergic neurons induce some prolonged REM sleep. These neurons are most active during REM sleep, and it's the pons projecting cells that seem to be sufficient for the effect. And finally, these are the people in my lab who actually did the work. Shinjae is the very first person who started working on sleep 4 years ago. Minshu got optical recording to work in my lab, which is terribly important. And so these 2 did the Basal Forebrain project with the help of Siyu. And Songhee and Johnny got rabies tracing to work, and Francis is the main person working on the brainstem REM project. And we have many collaborators. Thank you.
So you suggested that the circuit organization in the basal forebrain in terms of wakeful promoting cells is organized hierarchically. Does this hold true for the behavioral effect as well? So does for instance, the wakeful promoting effect of stimulating glutamatergic neurons depend on the PV cells?
So unfortunately, we haven't been able to do that experiment. So what you want to do is to activate the glutamatergic cells, but then inhibit the PV cells, right? So I mean, I don't know if you have a better trick, but you use Cree for everything. But then if you use Cree for one cell type, you can't use it for the other cell type. And so I don't know if there's a mouse with channelrhodopsin already expressed in the glutamatergic sauce. I don't.
Pharmacologically,
Right, so I guess it would be hard to target the PV cells per se, unless we have some kind of marker, alpha 5 or something that you guys have. But so right now we don't have the tools for that.
Very nice, very beautiful.
Thank you.
So I know it's work in progress, but I'm dying to ask, what's the connection of those cells with the basal forebrain and with Raphael?
With the Raphael?
Yes.
Um,
Do you see a connection?
So the anterior grade tracing that we've done is actually not synaptic. So all we know is whether the axon fibers kind of go to that region. We actually don't know what cell types they innervate, and we need a transsynaptic anterior grade tracer tool. So we don't know that. They kind of more or less project to that region, not very much. It's pretty sparse in that region. It's really going to the hypothalamus big time. And then the cholinergic cells project to the cortex massively. But by the time you reach the brainstem, it's kind of pretty sparse. So I think that to answer that question, what you want to do is use retrograde tracing. So you target the serotonin neurons, right? And then you see if you can see labeling in the basal forebrain.
And what about the septum?
Septum.
The theta generate.
The medial septum, yeah. So medial septum is actually part of the basal forebrain, not the part that we were targeting. We're mostly targeting the so-called HDB, MCPO, more posterior region. But these neurons project a lot to the medial septum. And so I tend to think of that as sort of almost like a local connection because it's all part of the same general circuit. So they do project there, but we also don't know what cell types they innervate there.
Maybe one last question.
I have a question about the GABAergic projections from the basal forebrain. So in earlier literature, using retrograde tracers in combination with immunohistochemical characterization of these neurons, suggested that there is a GABAergic projection from the basal forebrain to the cortex, which I think should be parvalbumin positive. I was wondering, what do you think about this all data? Do you think that it's wrong, or do you think that there's maybe like another?
So they do, even in our data, they do project to the cortex. It's just that it's much less compared to the cholinergic cells.
Okay.
So it's like we do see the projections there. And in fact, I just showed you the movie, but we have the quantification. We fitted everything to the Allen reference atlas, and we're going to put it online so everybody can just access. And you will see the number is not 0 in the cortex, but it's much less compared to the cholinergic cells.
Thank you.